So we have two professors from UCSF, Dr. Fischbach, Michael Fischbach. I opportunistically saw him give a lecture when he was receiving an award for his work on microbiome. And I thought he was fantastic. Dr. Fischbach is also a Hertz Fellow, which is a pretty big deal. And then we also have Dr. Wendell Lim, who is a Howard Hughes Research Fellow, which is even a bigger deal, I guess, to some degree, right?
It is.
No offense.
I just became Howard Hughes also, not that it matters.
Oh, you did?
I think we were an equally big deal.
I feel like I could tell you a few things about the work that they're doing, but I would probably butcher it. So it would be awesome if you could start off with an introduction on the kind of research and the work you do before you dive into the talk.
Cool, definitely.
Just so that we're all on the same page.
Fantastic. Okay, we're going to jump right in. We have a lot of ground to cover. This is going to be, first of all, thank you so much for coming. I can't believe you guys are all here. It's a little bit frightening and very humbling, but we're super excited about this. We've been talking about it for a while. This will work way better. I'm sure this will happen anyway, but just in case, if you guys jump in early and often with questions, and it's totally fine if we don't make it through the stuff we have. You'll see us skipping around on slides because we'd rather just answer questions and get into a discussion than run through slides. Okay? So, I work on the microbiome. Wendell works on mammalian cells. That's my introduction to what we do. You're going to see a lot more about what we do through the slides, so I'll save it for that, and you'll get a sense at the end of what we think is interesting. This is a very crazy place to start. Don't be frightened about the fact that we're starting on the carbon atom. This is the whole, you've seen these things before about the sense of scale, but this is a talk today about cells as drugs, and so it would help to contextualize where drugs have come from in the past and where they still mostly come from now, and why we think cells are going to be a big deal as drugs, okay? And why you guys may choose to get interested. So this is a carbon atom, as you can see, and then these adenine and methionine and glucose are examples of molecules. So most drugs are still chemicals, and they are, in terms of scale, about this size. If we move up in scale from molecules to proteins, and I'll contextualize this relative to a cell in a moment. The most interesting drugs that are proteins, there are two major classes of drugs that people use in the clinic right now. One is chemicals, which are sometimes called small molecules, and one is proteins, which are sometimes called biologics. And the major kind of protein drug is an antibody, and that's the size of the antibody relative to the molecule, okay? And then if we zoom out from there to see what size a typical cell is, it takes a little while before we get to a bacterial cell, which is about this size. Okay, so it's way bigger, you can see. And then if I zoom out even further, you'll see that the bacterial cell is way small compared to a typical human cell like a skin cell. Just a few microns across, maybe 3 microns by 1 micron, versus a typical human cell, which is about 30 microns. If a human cell were the size of a basketball arena, an NBA arena, the typical protein would be the size of a grapefruit, and the typical chemical would be the size of a grape. They would be very densely packed, shoulder to shoulder, no room. If you looked inside, you'd see no empty space. Any questions so far? The crazy thing about cells is the following. We like to use this analogy. If you have a bolt loose under the sink in your kitchen, using a chemical or a protein is like throwing a million wrenches into the house and hoping that one of them tightens the bolt. A lot of other things are going to happen too. Okay? A cell is more like a carpenter who could walk in, tighten the bolt, and then say, by the way, I'll stick around for the next 20 years, and if this bolt ever comes loose, I'll tighten it again. You won't even know I'm here. Okay? Any questions so far? So the crazy thing about cells that is going to make them amazing drugs is that they can go anywhere, meaning they can go to one place and not to any other place, unlike a chemical or a protein, which goes everywhere in your body if you have it injected or you swallow it. So they can go anywhere they want. They can do anything they want to, not just produce drugs themselves, which they very much can do, and they can make more than one drug if they want to, but they can also swallow things or build things. They can do what cells normally do in your body, which is the kinds of things you actually want to have happen if you want to solve a problem. And most importantly, they can make decisions. They can decide when they see something to not act, which is what they should usually be doing, or to do something different. Okay, so those things completely set them apart from all the drugs that you see on the market today. So far, so good. They are programmable. And as Wendell will explain more carefully, the degree to which they're being programmed right now, the alpha, pre-alpha versions of cell therapies that have hit the market, is almost laughable to you guys. And despite the fact that it's almost laughable, they are curing diseases that have been, somebody would be dead in 3 months, is now alive after 10 years and will die of something else. So already the horribly rudimentary versions of engineered cells are doing amazing things, and we think that the future will belong to cells that have a little more engineering than that. Yes?
When did they start engineering cells and using them as therapies?
The first cell therapy, I think it is safe to say, is a blood transfusion. That is, the cells being transfused with the blood are doing good, but that's not engineered. I would say that the first engineered cell therapy started roughly a decade ago. The ones that are and those Wendell's going to give a good background into. We still don't have engineered bacterial cell therapies in the clinic, but those are coming soon, and we'll talk about that in a moment. Yes?
What are the key situations in which engineering cells is a viable strategy versus where is it difficult right now?
You are asking relative to what diseases would you want to treat that way, or what cells would you want to engineer?
I guess it seems like the more direct question is probably what diseases you would want to treat that way, but probably that's a function of what the cells that you engineer for that situation.
We'll give you some examples, but let me, because I hate telling people to wait until later, give you one example now, which is that if you know somebody who has type 1 diabetes, they're constantly measuring their blood glucose and injecting insulin if they need it. The reason is that since the immune system has gone bad, they're missing a kind of cell called an islet cell in the pancreas that senses glucose, and when it senses it, it makes insulin. That's a very simple function, but there's no chemical or protein that knows when glucose is there and turns on. They're too small to do that. That's the perfect thing for a cell to do. Is I should be able to inject you with cells that will stay around for the rest of your life, circulate in your bloodstream, and replace what you're missing in your pancreas. That's a disease that's horribly managed today by current therapies because people still die in their sleep from type 1 diabetes. Nobody should ever have to die in their sleep or even measure their blood glucose.
Yeah, there's no question that drugs or molecules are much easier to produce in kids. There's been a 100-year history of how to do that. Cells are much harder, but that example of diabetes is just, we give someone insulin and we artificially create this open loop system that we have to monitor, and it leads to lots of problems. Whereas really what you'd like to do is have a smart system, closed loop feedback system that controls that in response to blood glucose.
Without you ever knowing that it's happening. Because that's what cells are doing constantly anyway in your body, is they're sensing stuff and making stuff.
That's that kind of intelligent, self-regulating system is the kind of thing where a cell is much better because it really is, a system that controls it. And then also the place where things are very useful are, very complex diseases like cancer, where you're talking about very subtle discrimination between normal cells and cancer cells. So there's a sort of, if in many cases, for example, antibiotics work great except for the resistance, right? And that's an example where a drug is perfectly fine with the limits of the resistance issue.
I have another totally random. Why does the size matter? You're emphasizing the size so much. What is it correlated to?
The size itself doesn't matter. It's the question of what scale does it take before you get something that's very much like Fantastic Voyage, like a robot running around making decisions. So the scale is meant as a way to set up this issue of what should be the agent of action if you want to cure a problem in the body. The agents of action, the molecules and proteins are what cells are full of. Cells are full of thousands of molecules and proteins that are moving around in a structured way to carry out jobs. it would seem to us that the way to solve a problem in the body is to use what the body normally uses to solve problems, which is cells. Everything happens at the level of a cell going somewhere and doing something. It's a whenever you describe things in terms of a network, you're doing that against the backdrop of what you think the system is. And I'm not using the body as a system, I'm more using, I think that the cellular network is the system at which most drugs operate, and they're usually doing something to break the network, and it usually has consequences outside of what they're meant to do. Whereas if you, cells are the most logical thing. If you flew in from Mars and say, here's a problem I want to solve, then the Martian would say, well, what normally solves problems like that in the body? And the obvious answer is cells. And the reason why they haven't been used up to this point in that way is because engineering them has been a little bit too hard. Does that make sense?
Yeah, I think those, typically you take this little tiny molecule and you flood your body with those little tiny spheres, right? And they go everywhere, and that's what the drug is.
Yes.
But what we're talking about is kind of this much more audacious goal of saying, can we take, relative to that, this big machine that does, can think and process information and make decisions, and use that as the chassis for carrying out therapeutic actions.
The challenges you'll see to preview it is how to engineer them, how to program them. And that, we're at, as I've said, a very rudimentary place with that. And it's going to be amazing. I think that the big advances, we both would say the big advances in engineering cells are probably not going to come from biologists. They're going to come from people who know how to program stuff. Because the biologists tend to do things in a one-off manner, and people who program stuff tend to come with a different approach.
What kind of cells are easier and more feasible to engineer?
So Wendell's going to tell you about a class, one of the two big classes of immune cells called the T cell. There are B cells and T cells, and the T cell looks like a fantastic chassis for engineering. And I'm going to tell you about some bacterial cells, any of which are reasonable to
But it
engineer.
would also be, stem cells, neurons, other, those are probably harder, and the applications are a little further down the line. But
Okay, I'm going to pause the questioning so we can move forward.
All right.
I love the questioning, but my handlers say that we need to move on. Okay, so when the PowerPoint fires up, so I told you, just in case you were wondering about the census scale, there are 10 billion proteins in one cell. So they're very densely packed, and that's the way cells are. They're fascinating. I don't want to knock chemicals and proteins too much, just so you know, here's maybe the most famous chemical drug, Lipitor, atorvastatin, bound to its target, which is called HMG-CoA reductase, an enzyme that carries out the slow step in making cholesterol. You make a lot of your own cholesterol, even though you ingest a lot in your food. So this interrupts your own ability to make your own cholesterol, and this drug is probably, it is estimated, saved more years of human life for people not getting cardiovascular disease than any other drug. That's a chemical, a protein that many of you may have heard of. I showed you the antibody molecule, which looks like this, but has a stalk, which would come down here, and a second arm, which would be over there. This is one arm of it bound to a marker on the outside of breast cancer cells called HER2, and this is Herceptin, which many of you may know has saved many women who have breast cancer. So nothing wrong with chemicals and proteins. They've done tremendous things. It's amazing, in fact, how many diseases can be cured or managed with by flooding your body with a chemical or a protein. We'll go into many others that are not well managed by any chemical or protein for reasons that I think are obvious. And again, because that can sound cryptic, one thing that's not good to do globally all over your body, for example, is to suppress your immune system or to activate your immune system. There are a lot of drugs right now. There are diseases that are treated by globally suppressing your immune system. That's a bad idea, because you might solve the problem you're trying to solve and then create 3 other problems, which is what people see in the clinic. And same is true for globally activating your immune system, which might help you deal with one problem, but then you'll have autoimmune disease. Okay, so far so good.
Actually, I have a question on that.
Please.
Given that thought, and earlier slides suggested that we would have these cells sort of flooding throughout the body, cells being the clinically divided ones. What about localizing those cells?
But perfect setup for engineering cells so they go to where we want them to go and not where we don't want them to go is the big thing Wendell's done over the last couple of years. So he's going to tell you exactly that. Was that what you were asking?
It was. I'll follow up later.
Okay. It's a critically important question because one of the major benefits of cell therapy is just that it could act in one place in your body and not elsewhere. That's just a mind-blowing thing relative to the way that flooding your body with a chemical or a protein works. This is how big bacteria are in the head of a pin. They're all over the planet, but okay, this is my 5-minute introduction to the microbiome. So you guys have heard, maybe some of you, about the microbiome, but we have a lot of bacteria growing on us, and they're all over the body, but the largest numbers of bacteria are in your gut. I try to understand systems in an unemotional way, and my unemotional description of the microbiome is like this. You cannot avoid having bacteria present on all surfaces, on the walls and the floors of the room, and all over the outsides and insides of your body. So the way that multicellular organisms have dealt with that is to co-opt them. And the way we've co-opted bacteria is that we have devised a way of feeding ourselves, of extracting nutrients from food that takes advantage of the bacteria doing it. In fact, the bacteria eat all of your food first, and then you eat their bus exhaust. Okay? So the inside of your gut, it's full of bacteria, and these are the two reasons why. Bacteria are actually fantastic at pulling nutrients out of food. It's a great system. It works really well. This is why they're there. They're there because you couldn't avoid it, but also because we work with mice that don't have any bacteria living on or inside of them, and they're very sick. They don't pull nutrients out of their food well. They live in a bubble. Okay, so here's a few things that they do. When food comes down the pipe, they multiply and they get the first chew, and then nutrients go into you. We'll talk later about the fact that even that simple process, the fact that you eat a steak and a salad and I do, and then some of that mass becomes more bacteria, which is a larger stool for you, and then some of that mass becomes more you, that the ratio between how much of that goes to more bacteria and more you is not the same from one person to the next. And a very simple thing one could do if they wanted to change the amount of weight they gain when they eat is to replace the community they have with one that's much more stingy about keeping calories to itself. Does that make sense? It's very, very simple things, but a powerful concept, I think. The other thing is that the way the system is set up, there are sensors in your gut that chemically sense things in food. We don't really know exactly what yet. When those sensing events happen, they signal directly to the brain. This is through hormones. And then the brain will tell you, should you eat more or should you not eat more? If you were living in the bush, this would work well, and you'd get hungry when you should be hungry, and then you wouldn't eat when you shouldn't be eating. And these signals have gone a bit awry in places like here where there's abundant food. Knowing what the signals are would be quite helpful, and we think that many of the signals are actually coming from the bacteria. As they digest the food, they're generating signals that tell you to stop eating, and if we could co-opt that circuit and restore it to the way that it's supposed to be in people who don't eat too much, that would be helpful. Okay? So this is a lot about metabolism. I'm going to tell you about the immune system real quick, and then this gets to the where I'll stop, which is in saying how we could engineer the system and what the goals would be, what diseases we would want to treat, and how we would do that. Okay, so this idea that that bacteria multiply rapidly when you eat is you don't appreciate how unstable a situation that is. I didn't say this in the lecture because it would have been too morbid, but if you die and I let your body lie down on the floor here, then it wouldn't take very long before the bacteria that are in your gut would chew through you entirely. Like your eyes would be gone in a few days, and your nose would be gone in 2 weeks, and so they'll eat you up. Okay, so they're helpful under normal circumstances, and you should think of them that way. But there's a very active process by which we have to push back against them to prevent them from taking over. It is not something you should take for granted that you have this huge bacterial cell division multiplication event happening in your gut every time you eat. That's being kept actively in check. Right? Does that make sense? And so that's generally the job of the immune system. Old view is that the immune system's job was to differentiate human cells from everything else, including bacterial cells. And that should be an easy thing to do because bacterial cells look a lot different from human cells. New view, which is the right view, is that in fact the immune system has a hard job to do because it has to tell the difference between the good actors, which are human cells and helpful bacterial cells, and then the bad actors, which are also bacterial cells and often look quite similar to the good ones. That's a much harder thing to do because you can't have an autoimmune response against the helpful bacteria. That could cause a problem. Does that make sense? So the corollary of that, what has come out recently, and I'll sort of cut to the chase and flip through this quickly, is that the immune system does something that we still don't understand fully when it encounters any bacterial cell, potentially helpful or potentially harmful, in which some subset of the bacteria are completely engulfed, split up into their little constituent fragments, and then those are sniffed by the immune system. The immune system knows when it sees those, whether it's found a bacterial cell that it doesn't like, in which case it mounts an immune stimulatory response, and a cell that underwent that sniffing event will become a pro-inflammatory immune cell. Or, and this was the new part that wasn't known until recently, an immune cell can encounter a helpful bacterium, and the same naive immune cell can become an immune suppressive immune cell, which is this, if you can believe it, brand of immune cells wasn't even known until a few years ago, called a regulatory T cell. The amazing thing is that it looks like by putting in a specific cocktail of bacteria, by choosing which bacteria you put in carefully, you can program the immune system to be much more immune suppressive, which would be great for somebody who has autoimmune disease, or immune stimulatory, which in some circumstances would be great for people who have cancer and need more of a high octane immune response. And the ability to program that through bacteria, which would be much more specific than just giving you a drug that amped up your immune system all over your body, would be exciting. And Wendell's going to get much more into turning the dials in the immune system.
Is this the trade-off? We only have so many undecided cells.
It is a trade-off, meaning that the more immune suppression you get, the harder it is to do immune stimulation and vice versa. It's a false choice, though, in the way that it's been pitched clinically, in that the way it's done right now is you're either giving somebody, the term is parenteral, it means system-wide immune suppression or immune stimulation. What you really want is to be able to say, this person has melanoma, I want to specifically stimulate the immune system in the skin and leave it the same everywhere else. Or this person has an autoimmune disease like inflammatory bowel disease in their gut, I want to specifically suppress the immune system there and leave it the way it was everywhere else. So it is a trade-off in a tissue-specific way. It doesn't need to be a trade-off system-wide. Yeah?
You said the immune system has a tough job deciding between good and bad
Yeah.
bacterial cells. Is there evidence that some bad bacterial cells exploit the undecided cells and suppress the immune system, even though it should be the other way around?
Famously. Yeah. They're just people who spend their whole career studying that. It's fascinating stuff. It'd be great to turn some of that to our advantage in being able to trick the immune system into mounting a pro-inflammatory or anti-inflammatory response against some tissue in the body, which is something that's a broad family of outstanding challenges, a broad set of diseases that are not well
And I'll
tolerated.
mention how some cancers do the same thing. They kind of harness the machinery that leads to immune suppression, so they create a local suppressive environment.
Yeah, in fact, they can't become cancers unless they do that. That also has not been realized until recently.
Is there a global understanding of you mentioned there was some local targeting that can happen where you have something in the skin and then an immune cell comes along there, it finds something happening in the skin, some other guys come along and they join the party. Is there some global understanding that this is happening in the skin in this area?
Global understanding by the rest of the immune system?
Yeah.
There is. The immune system is very skilled at signaling with diffusible chemicals to each other. So when somebody encounters a problem, one of the first things that happens is sending out signals to its buddies to recruit them. Or when one of these immune suppressive cells comes, it sends out signals to stand down to everybody in the surroundings. So there is a sense in which that happens, and then the way that immune cells are trained in the first place happens in the thymus, and so the immune cells are like constantly being trained one place and circulating and going to find trouble or to stop trouble elsewhere.
Does this mean that if we make a certain area suppressant or stimulant, that the rest of the body goes to that, the rest of the body's immune system goes to that area?
You'll get, the rest of the body will see that area, but there's a sense in which, how would I explain this to you guys? Immune cells are acting like logic gates. They take multiple inputs and then they make a decision about what they should do. It's a fantastic thing. It's exactly what they should do. One of those inputs will be markers of what tissue they're at. So if they see what's happening in the skin, but then they go to a different tissue, they could see all the rest of their inputs to their Boolean logic gate could be the same, but the fact that they're in a different tissue, they'll respond differently. So that's good. That's what we want. Things that are used right now are acting like a sledgehammer. They're just saying, forget all the other inputs, forget the circuit entirely, just turn on. It's a totally crazy approach to regulating the immune system. The immune system already does a fantastic job. Sometimes one little thing goes wrong and that causes autoimmune disease or something like it, and the way to fix it is not by pointing the sledgehammer in the opposite direction.
I think one answer to that is that it depends, that immune cells do have ways of communicating with one another, and some of them have different length scales. Some are very local, some are very long range, and so you can in some cases get very local effects, and in other cases you get very sort of systemic ones.
Okay, so probably the thing that I would be most excited about up front with engineering the microbiome to do something, because it's the most concrete thing we know of thus far and it's going to work, is in the way that the bacteria that live in your gut make chemicals. They make tons and tons and tons of chemicals. This doesn't come as a surprise to somebody like me, because I grew up studying how bacteria make chemicals that are used already as chemical drugs, a lot of antibiotics and anti-cancer agents. The big thing that we've found in the last few years is that the bacteria that live in the gut make tons and tons of chemicals, but that the list of chemicals that are the most abundant in my gut and the ones in your gut are completely different from each other. This is a huge, I think, entirely hackable difference between me and you. We're still sort of in the process of figuring out just how much I differ from you, and then what would be the optimal cocktail to have. But this is, especially in light of how similar people are at the level of the human genome, a stark difference from one person to the next. Okay? Yeah.
Does this mean that, so if I have a different population of bacteria in my gut, maybe it would be producing different types of hormones or other things?
Absolutely.
Okay.
Totally. And to give you a sense of who's there, there's a lot of confusing stuff you might read in the news. The way you should actually think of it is that the population of bacteria that live in your gut is simpler than the narrative you've been sold, which is hundreds and thousands of species and stuff. It's not like that. It is about 10 bacterial species make up 90% of the cells in your gut. Then there's a long tail on the histogram, and a bunch of low abundance guys, some of whom are probably doing important things and others you could do without. Okay? The other thing that I'll mention now, even though it comes up later, is that this system is completely changeable. What we've learned from this big human experiment with fecal transplants, some of you have read about this, people get really nasty gut infections, and now they'll get a fecal transplant to correct them. That had been done for a long time, but had never been done in an organized way, and about 4 years ago now, a group in Scandinavia finally did the kind of gold standard clinical trial, a randomized controlled trial for fecal transplant. It took them a long time to get this published because nobody believed it. But so what they found was that a fecal transplant for this really nasty infection worked 90% of the time, and the drugs, the chemicals that were used before that, the antibiotics, worked 30% of the time. So like the graphs in this paper were just astonishing. Yeah, I may even have put them in. That was, in my view, the least surprising of the things they found. The more surprising thing they found was that it's totally safe. People simply do not see any adverse events from. What would happen is you'd get. Have you ever? Nobody in here has done a colonoscopy prep before because you're all young. You have. I have too. So you wash out.
Yep.
Then you would. You can now just do it with capsules. You would swallow like 20 capsules and of something that of feces that's just mashed up in salt water, and you'd be good to go.
How does it replace your existing system?
Yeah, Ruchi was asking a question first before everybody.
Well,
Yeah.
we heard salt poop pills.
I'll get to that.
I was just wondering, how does it? What do you do with your existing? You can't take everything out, right?
You get to the point by doing a colonoscopy prep. You and I've been through this, so you know what it's like, where you've washed out most of what's there, and then the new guys you swallow are in such vast excess to who was there before that they become the king of the hill. And because adult human bacterial communities, you guys are all old enough to have, in fact at age 4, you're old enough to have a bacterial community in your gut that is quite stable, that doesn't change much over years. It's just ecology, it's the founder effect, it's stochastic stuff.
Unless you have a bad case of food poisoning.
Unless you take antibiotics or get food poisoning, in which case.
When you take antibiotics, sorry, what percent of the bacteria in your gut, maybe we don't know exactly, are wiped out? Like, can you wipe everyone out and then?
No, you never wipe everyone out, and it's typically inconsistent what happens after that. Sometimes you return to a state that's similar to where you were before. Sometimes you end up in a completely different place.
Interesting.
Yeah.
When you get a colonoscopy prep, does it remove enough of the bacteria that whatever you ingest kind of next ends up becoming dominant?
Well, if you take poop pills, they definitely become dominant.
But why is
There's
that
a lot of
standard?
bacteria.
But that's not a standard procedure.
No, if you don't, like the colonoscopy preps I've done, I haven't taken anything, and then
Next
what?
thing you happen to eat.
Well, it's a competition between grow back from the bugs who stick to the mucus in your gut, and whatever is on. If you were on an organic farm and they didn't wash their lettuce, so yeah.
Mike?
No, oh, sorry.
Oh no, I was just curious, do we understand where most of the variation comes from? Is it food, environment?
It is somehow you're encountering them. I don't think we know exactly, but I have kids. Everybody's got little kids. They're just, there's no problem with encountering stuff. They're like licking and putting everything in their mouth.
So there's not really a genetic component to that?
No, there's essentially no. It's been surprising how essentially there's no human genetic component. And so I was going to say, completely safe, and also that if I give you a fecal transplant, it engrafts is the term, if you can believe that, essentially 100% of the time. So there's no special community for you. I could turn your community into anything. Anybody in here could be a donor for anybody else. There's no need for a relationship, which makes this system totally happy. Whoa, that's like 5 hands went up. Yeah. So.
How differentiated are the outputs for particular sets of microbiomes? So, meaning, can different microbiomes produce the same outcome?
Different microbiomes can produce similar outcomes. Very similar microbiomes can produce surprisingly different outcomes.
Why is that?
That is because closely related bacterial species can do very different things in terms of what we feel, making chemicals, suppressing or activating the immune system.
If
That's
different species can do different things, why is the risk low? Why isn't then some of the species doing something
The risk,
crazy?
when I say risk, what I should qualify that to mean that the risk of having like a crazy immune response and is coming to having to go into the hospital is very low. You're undoubtedly changing your biology by a lot by doing that, and that
In some way.
in some way that we're still yes, exactly. Oh geez. Yeah.
So you suggested that things like weight loss, weight gain, for example, can be dependent on your bacterial population.
Yeah.
So then, if the bacteria can change in a drastic way when you do a colonoscopy or take antibiotics, why does that not change like randomly, like people who would tend to get start gaining weight after taking antibiotics? Does that happen?
That actually does happen. There are enough anecdotal reports of things like that the doctors believe it. So, and we're talking about effects that it's not going to be like one day you wake up and you've gained 20 pounds.
Right.
These are soft effects, 10 or 20% effects. At 10% effect, if you're gaining 10% more weight, you'll notice that 3 months on, and it's a soft thing. Hugely important for your health, but it's not the kind of thing where you'd be like, holy shit, it was that colonoscopy, and you know. But for people who have are trained to look, they have Crohn's disease, there'll be, you know, there are tons of anecdotal reports which are now sort of essentially believed to be fact that that a flare will come often after a course of antibiotics. As you,
So why is there no skinny boot clothes?
people are evaluating that right now. There was a hand up over here.
How similar is the bacterial profile between donors? So if you have, like, say, person A and person B, and you do a fecal transplant, is it you end up with the exact same like population levels, or is it just rolling the dice?
You end up looking like the donor.
Or do you, like, exactly, or do you end up with like because you're coming into a pretty much empty system, right?
Right.
Do the bacteria always end up equalizing at the same level?
Yeah, so in the experiment you're asking about, what you really were asking was if you take a mouse that has no bacteria in it at all, and you take a population of 10 different bacterial species, and you just randomize the relative levels, do they always turn out to be the same once they enter the animal? Is that what you're asking? And the answer is that they always seem to even out at the same proportions. The modifier would be if your diet were totally different from the donor's diet. If you were a vegetarian and you got a donor kidney from somebody who eats meat, you'll lose a bunch of bacterial species whose presence is associated with chewing on meat.
Do you need to wipe?
Wait, Aaron, go first. You've been trying to
We don't necessarily understand the full impact of all these bacteria. So we swallow the poop of an Olympic athlete, we think we're great, but there's something we don't understand.
Right, and that person could have been eating a wonky diet, and that could be a little weird. I have no idea.
Or it had there's no environmental impact somehow to your biome. It's only
The environment initially is going to determine what you encounter and who sets up shop there for a kid.
right.
But once you've got bacteria there, the major environmental impact on who's there is what you eat. That's essentially the only thing that matters.
Not smog, not Beijing, none of that.
None of that.
Because it doesn't get into your intestine.
Exactly.
When you,
No, use this perfect. This is just what we want.
when you already have a population of bacteria,
I'm going to finish it in 1 second. Yeah.
when you already have a population, to repopulate, do you usually have to wipe out the existing population? Or if you kind of slowly change your diet, does your population also slowly change?
If you slowly
And it
change
becomes
or
completely
quickly change your diet, the community will change in a way that's reversible. This experiment was done. Has anybody lived in Boston before?
Yeah.
Have you ever read Red Bones? Red Bones, some of you have, yeah. So a colleague of mine did an experiment where he took a bunch of people and he split them in half, and half of them had to eat all plant diet for a week, and the other half ate all red bones. Morning, noon, and night. All meat for a week, and then they went back to their normal. So it changes dramatically during that time, and then it goes back.
To, given your normal diet. But you still need to wipe it out completely in order to repopulate with like the...
If you want a total repopulate, if you want to do the transplant, you need to drive out who's there, because who's there is going to be itself a stable community, and typically the king of the hill dynamic is that whoever's there is really hard to bump off.
So there's like some distribution of stable communities that exist.
That's right, and we don't understand what they are, but every one of us is populated by one of them.
Presumably there's sort of these stable attractor states,
That's right.
and that some perturbations can push you off.
Got it. Like,
Can you tell if the bacterial population has changed through some of the things Kanjan is talking about through your poop?
If you look at your poop and it looks different, you're asking, does that, if just by visual inspection, or you sometimes can tell. Although the things in poop is almost all bacterial cells, though you do see the totally indigestible stuff in there. These are the parts of the plant cell wall that can't be broken down. So sometimes if you're looking at a color change, it could just be that you ate something really
no, but
hardy.
I mean changing poop over time.
Yeah, so changing poop over time is probably a readout of a change in your community. Although I don't know of a study. I can informally tell you that I've seen that. So,
I've seen it. So that's another validation.
in pregnancy, it goes way off the rails in the third trimester. Way, yeah.
When you say poop is almost all bacterial cells, like just all the poop that you poop, does it like grow like after you eat, like within the digestive cycle?
Oh yeah, yeah. There's a huge,
Interesting, yeah.
there's a huge bloom. You eat and then,
That's fast.
yeah, oh yeah.
I think I missed this.
You and then I'm gonna finish and then Wendell's gonna start.
A lot of questions.
No, yeah,
I think I
gotta be
missed
good.
this, but if when you do the fecal transplant, and then let's say you stay the same diet, and I'm guessing there's some period, let's call it a month, when everything switches back. What was the virtue of doing the fecal transplant?
Well, so fecal transplant these days is done, but this guy's got something to give.
Thank you.
Yeah. Christmas in August. The virtue of doing the fecal transplant these days is that unless you're in a clinical trial, the fecal transplant is being given to you because you have a bad infection in your gut that's really hard to rectify with antibiotics. What will often happen is you'll take them and you get better for a few days or a week or 2 weeks, and then it just comes roaring back. You have to go back into the hospital, and people go through cycles of this. Sometimes they'll die, but often they'll just go through like a year of going into and out of the hospital. So the virtue of getting the fecal transplant in that case is, and it's funny, anybody in the room with a healthy donor community could be the donor, and it would get this person back to a healthy state.
Okay.
So that's the only way it's used right now. People want to use it for all sorts of stuff, but right now they're not engineering. So this gets me the last thing I want to say. I'm so happy you guys have. Yeah. Which is, there's two ways to engineer the community, and they're both interesting. One is by engineering bacterial cells to either make or not make things that they originally made. One of the most obvious examples of this, and I encourage you to look it up if you haven't read about it, is this molecule, this very, very small chemical, even relative to other chemicals, which it has the long name trimethylamine N oxide, short name TMAO, has gotten some press appropriately because it's made exclusively by bacteria in the gut, and it comes from choline and carnitine, two things you may have heard of which are in food. They're especially in meat and eggs. There was a guy who's a friend of mine, I was in fact just seeing him earlier this week, who is at the Cleveland Clinic. His name is Stan Hazen, and he did something you would have thought would have happened a long time ago, which is he went looking through blood to find if there are any chemicals whose levels would be better predictors of cardiovascular disease than what doctors currently use, which is a test called C-reactive protein. And he found several chemicals. They were all exclusively from bacteria, which is crazy. And then the top one was that molecule called TMAO. Bacteria in the gut convert choline and carnitine to this, and then in the liver, this becomes this. Okay? And this molecule, its levels essentially predict without any doubt whether you'll get cardiovascular disease and how quickly you'll get it.
When was this?
This was only a few years ago.
So do they measure it for everyone who's at risk for cardiovascular?
That test is just coming online. They do it at the Cleveland Clinic now, the biggest cardiovascular disease unit in the country, but everybody else will catch on soon.
Does anyone repopulate the bacteria of people who have high levels of TMAO?
Meaning, has anybody tried to come up with a therapy to replace a high TMAO community with a low? Yeah. No, but that's something we're very interested in doing. You can imagine that so nobody thought that the microbiome, even a few years ago, would have any connection to cardiovascular disease, but this data is so stunning that it's a...
Can you commercialize any of this research?
Yes, you can, because it's... So
like tomorrow morning, maybe?
Yeah.
General question is, like, you talk about the timeline here?
Yeah, actually.
Little context from your perspective on the timeline of understanding, of research, of commercialization, of...
So right now, the level at which people are operating is that they are figuring out basic stuff like this, mechanisms, and and companies are doing very crude things, like just trying to use fecal transplant for everything. Oh, I wonder if we just randomly do fecal transplant, we'll end up curing some people of fill in the blank in almost any disease you can think of. The prize, in my view, is going to belong to the group that figures out how to do synthetic ecology, how to build a community from scratch that does what you want it to do, that is stable, that makes what you want it to make and doesn't make what you don't want it to make. So Wendell and I will both tell you that we both have commercial interests. Wendell has set up a company. I'm in the process of setting up a company to do stuff, and there is a huge amount of academic work that needs to happen because there's just so much stuff, as you can tell, that it isn't known yet. So a therapy that addresses the TMAO problem is probably 3 to 5 years out. People still don't know how to build bacterial communities that are going to do what they want them to do and behave well in a patient. But I think the first ones will look rudimentary and won't work as well as the next ones will.
And does this kind of fecal transplant also require FDA approval, like regular pharma,
Yes.
chemical, molecular drugs?
It does, but the FDA approval route has been really easy because so many fecal transplants have been done in the safety. So usually when you develop a drug, you worry about efficacy and safety is a greater concern than efficacy at a company like Genentech. These are so safe that FDA has become quite permissive in allowing you to just concoct a cocktail of bacteria and give them to a person.
So
Why?
Yeah.
Why?
is that
Why
expedited?
Yeah. What?
I don't know.
Okay.
Sorry, is it fast track? Is the FDA approval?
It's not
fast
formal
track
fast track, but it goes through a different office than it's the office that handles protein drugs and viruses and things like that. So it's not you can get a fast track designation if you're going after a really difficult orphan disease, but it's just they basically say as long as you're using commonly occurring bacteria in some cocktail or even engineered bacteria, it's going to go fast.
Hey, is there a mapping of bacterial group distribution across populations yet?
Spatially? I mean, geographically?
Yeah, you look way different. I mean, I think that the mapping is more by what you're eating and encountering.
Right, so I mean, is there one that's in real time now for...
No, not in real time. No.
Nobody's doing that, right?
No, but people are right now just beginning to go off into the bush and find people who haven't been...
But that data seems somewhat useful.
Oh, hugely useful.
But are we aggregating this data across population in real time, like Ubiome and stuff? But is that a
There's a big project called American Gut that's trying to do it in the U.S. and there are companion projects to try to do something similar in the EU, but it's not really. You wouldn't call it that well organized. The American effort is somewhat well organized, but of course it's just limited to the U.S.
What's your opinion on like the whole crazy thing about probiotics? It's like,
I think that it's a huge hoax, but that it's a hoax that works for some people. So like most people, a healthy person goes to Whole Foods and buys a probiotic. It's not doing, I don't think, anything for you. There are ingredients that they put in there in some probiotics, like fiber. The fiber might actually be doing something for you. There are people who have IBD for whom immune suppressive drugs do nothing, who swear by probiotics, and I actually believe them that in their case this might do something totally unproven, and it's crazy how much people spend
I
on
will
it.
say for pregnant women, probiotics work. It does help you poop.
Okay.
But it might be the
Whatever
fiber, it might be there.
Whatever it is,
Yeah.
it works.
Okay, don't stop taking them. Just know what science is behind it. Yeah.
So can you talk a little bit about the mapping? So what does it come down to? You sequence a bunch of microbiomes across the world, and you assume that one of them is good for you versus others? Or how do you get from, I have a bunch of microbiomes, I'm going to sequence them, and then this one is actually going to do something?
I'm on more the experimental side. The sequence analysis to know who's there is critical, and that's been fairly well worked out. What's harder to do is to take a community of bacteria and then say, here's an experiment I can do with it to tell me whether it's good or not. Two things you can do. One is you can just put them in a test tube and give them a precursor chemical and see what they turn it into, and that's a really predictive experiment, meaning that if you see something in the test tube, you'll see it in an animal. And the other is that you can take an animal and either do a fecal transplant to it or take one of these special animals that have no bacteria living on or inside of them and then populate that animal and put it through some kind of test to see a health benefit or detriment that the bacteria confer on the host. So those things are still happening really little, infrequently compared to the amount of just mindless sequencing that's going on all over the place. There's a lot of over-interpretation of sequence data and under-checking of what it actually means. Last question, and then it's Wendell Street.
Is what what you're proposing to engineer the actual bacteria in terms of what they produce, or to engineer a community of bacteria that you would then populate, like a stable community controlled for various factors, that then you would populate with a bunch of people instead of taking like random fecal samples from random people who may or may not have different problems?
Yes on both. A big yes
so you
on
also
both.
plan to engineer the bacteria.
Yes.
Is it possible that that would make the therapy less safe?
It is possible, but there are easy ways to address that.
How so?
First, deleting genes in bacteria rather than adding genes is probably a safer thing to do. And then making sure that if you engineer bacteria to make something, that you put a kill switch in, maybe a doubly or triply redundant kill switch, is a good idea. A question that's relevant to engineered human cells too. This is awesome. I'm totally stunned. This is so awesome. But let me turn over to Wendell.
Wendell, there's a bottle of water for you.
Oh, that's
So John, you want to move here, or are you good?
You get out of your way.
Okay, so what I'm going to talk about now is the idea of engineering or hacking your immune system. And let me just start by saying, why are we talking together? And we're both really enamored of the power of cells as opposed to molecules and their complexity, and want to harness that. But I think one really common thing between the microbiome and the immune system is the microbiome is really, we're realizing, it doesn't come from you, but it's a piece of you, right? And it's an important thing, but it's a piece of you that we can kind of access and change. And so we think it's a really good way to interface with your body, okay? And I'd say the same thing for your immune system. Your immune system is a sort of a set of cells that we can actually access and we can change and put back in you. And so it is also a really powerful, it's a semi-autonomous system that's really, I think, another powerful way to interface with your body and disease. So I'm going to talk about engineering mammalian cells, particular immune cells, but I just want to point out that the sorts of things that we're trying to do and we're thinking about, you could do with stem cells to help repair tissue, with neurons to help sort of change connections in the brain or other things like that. But really kind of the lead here in today's world is the immune system, because it is so accessible, as I said, and that there's a real clear application for which immune cells can provide really big advances, and that's in treating diseases like cancer. So many of you probably heard about cancer immunotherapy. There's just some great articles in the New York Times last week. This is a really hot area, but I want to point out that it's really just it's the lead in what we think is a bigger movement to be able to engineer cells and take advantage of the power of cells. Okay. So if there's one thing that I want you guys to take away from this, and you may agree or not agree with me, but I'll tell you this is my thought, is that we think that engineering cells, particularly immune cells, and the challenges of trying to treat a disease like cancer with that is really has a lot of the same challenges of some of the sort of algorithmic challenges that you guys might face in things like designing self-driving cars and algorithms for that, as well as things like face recognition. That it comes down to we need to be able to engineer cells to be able to really look at systems in the body, know where they are, be able to recognize things, and then only under very specific conditions execute some kind of program. Okay, so you can tell me if you agree or disagree after we talk about this. Working. Okay, so let me try to step back and introduce you to the immune system. So the immune system is a set of semi-autonomous cells that patrol your body for foreign cells. So this is one type of white blood cells, part of your immune system. This one actually is responsible for eating bacteria that come into your body. So that's a little bacteria there that it's chasing. And so you can see, this is a piece of you, but it really is this self-acting amoeba-like cell that literally smells molecules coming off that bacteria, chases it, and then eats it.
You were wondering how that was going to end.
Yeah, wait, how do you record this?
This is a movie from the 50s. It's like the best movie ever. It's like it's really dramatic. There's actually one case where it sees another cell and sort of splits its head. But they it just shows the power of cells. They really can do very complex functions. This is programmed to smell out the bacteria and eat them. Okay. Now,
So imagine what's involved in that. You need to have your smeller on, and then you need to be smelling in various directions, and wherever you're smelling most, then you need to reorganize cell movement, which is a huge undertaking towards where you're smelling the strongest smell from, which is that's a really hard thing.
And I think that's a good place to take it. These cells have sensors, just like, and they're designed to sense various things like the bacteria. And then there's sort of an information processing layer that then goes into engage this molecular engine that leads to that. Your question? Oh, yeah.
Is there any idea that they're communicating at all? The cells are communicating at all there?
Which cells of the immune?
So there's a few different cell types I see in there.
Yeah.
Ignoring the bacteria for a second, are the cells at all communicating with each other?
They are. They're always communicating with one another. These are red blood cells. They actually don't communicate too much, for example, these cells will communicate to other guys like them, their brethren, and call them in.
Right, I just see it in that. So I was wondering if when the guys move out of the way, if they're just pushing them out of the way? Or
Yeah, these are red blood cells, so they're relatively, their job is to carry oxygen, and so they're relatively inert
They're
in
the
terms
exception.
of.
If there had been more other immune cells in there, you would have seen some crazy stuff. They were left out just to make things
Right, but when you can see movies where like someone gets a cut or infection, and then these types of cells will just kind of come pouring in. It's really quite amazing.
For those of us who are dumb about cells,
Yes.
is cell mobility difficult? Like that thing's moving around what seems like relatively quickly. Cells
yeah.
just do that easily, or is that
This cell is designed to move very quickly.
Right, so the cell has to be particularly.
Yeah, but literally a lot of the molecules in here are the same as the ones in like an amoeba out free living in a pond that's doing stuff. So it's just so it's sort of a convergent evolution that this came from you, but it's like an amoeba, and its job is to really wander around your body and patrol it and look for trouble. So the
You're asking how hard it is, just it's not like having an outboard motor and then you go.
That's
you
right.
have to reconfigure all the struts inside, all the structural things as you go. They're constantly being broken down on that side and built up on that.
It seems great. Yeah.
Very
it's kind of a swarm thing. It's it's the way that a lot of things work in nature. I wouldn't say it's that hard.
Cell communication. Just do most cells communicate via the same mechanisms, and if so, what are the
with each other? With each other. I mean, they can touch each other. I mean, so they're all based on molecules, right? And you have these sensor molecules, and they could touch another cell and touch and recognize molecules on that cell, or they could recognize molecules that are secreted and are diffusing, and those could come from far away.
I think what you're asking is the language common between immune cells and structural cells?
Is there a protocol? Yeah.
They have channels for communicating with one another.
The language is common.
Yeah.
What is that? What is the
language? Is chemicals and small proteins.
It's molecules that recognize one another, and then those trigger downstream events in the cell. Okay.
Are they like mostly proteins and protein receptors?
Yeah, it's mostly proteins and protein receptors. But one key thing is that a lot of that is very modular. That one sensor in this cell type might trigger a certain response in cell type A, but be used in a different way in cell type B. Okay.
Neurotransmitters are used in the brain to do the things and they're also used elsewhere in the body to do totally different jobs. Same sensor apparatus.
But I'd say this is a common theme here that we're going to close with, is that idea of what is that language and how do we actually that evolution has used, and how do we actually learn to program in that. So getting back to this, this shows a cell that is designed has evolved to kill foreign bacteria, but we have other cells that kill other types of foreign cells. For example, if you have a cell that's infected by a virus or a cancer cell, there are other types of immune cells, particularly these things called T cells, that have the ability to recognize and kill abnormal cells. Okay, and this is really the one that we're working on the most with respect to cancer. Now, an important thing to remember is that you can focus on the killing function of these cells, and obviously that's really, really important. But what's probably even more important, and more of the genes in these cells go into trying to tightly control that decision, because as you can imagine, if these cells, these killer T cells, if they went around and killed many of your normal cells indiscriminately, that would be very, very bad. And in fact, that is what autoimmunity is. It's poor diseases that lead to poor regulation, whereby these cells make bad decisions. So they're very tightly controlled. There's a lot of gating that controls whether they'll launch this kind of killing function. Okay. They've really evolved to carry out sophisticated recognition and response algorithms. So cancer in the immune system. One thing I want to point out is that there are things that are protocancers are coming up all the time, abnormal cells from mutation. And most of the time they are eliminated by the immune system, okay? That is, they're recognized as being funky, and these T cells will mount a response and kill them. Okay, but you know that you can get sustained cancer, and a sustained sort of manifestation of the disease occurs if one of these two things happens, or probably most likely both of them. One is that there's a problem with the cancer cells where they're not recognized as foreign. And that's really not that surprising in the sense that cancer cells come from our own cells. So the idea that there would be some clear marker or molecule on them that would be distinguishing is not necessarily true. So they can be very similar, and so a T cell can have a hard time recognizing it as foreign or not, or abnormal. The second thing is, and we talked about this before with Michael, is that often as cancer really is cancers evolve, and one of the things that successful cancers do is they start producing things that are immunosuppressive. So they can block the activation of these T cells. So even if a T cell could come in here and recognize this, it might be suppressed so that it couldn't do that. Okay, so with the recognition of this, that's when we see in the last really 5 years or so this explosion in what's called immunotherapy. And one thing I want to point out is, although they involve this whole system, there are really kind of two really different flavors of immunotherapy out there today. One is called checkpoint inhibitors. These are antibodies that actually block the molecules involved in suppression. Okay, so they prevent the things. There are certain cancers that we know produce certain signals to suppress, and you can put in antibodies that will block that. And what that will do is, and this is where you see these people responding and actually getting cures. Jimmy Carter was an example with melanoma. He got cured. Now, interestingly, when you give someone a checkpoint inhibitor, there are about 20% of people who respond. They can respond in we're talking about responses that are essentially cures as opposed to most other drugs that are extending life by a few months, okay? But an important thing is with checkpoint inhibitors, the response rate is about 20%. So about 80% of the population does not respond. Do you guys have any idea why that would be? If you're blocking this immunosuppression, what do you think? There's two sides of this equation, right?
Maybe they're already immunosuppressed?
Well, they are immunosuppressed, but let's say that that is a problem. What could be missing in those 80% of the patients? Yeah.
Maybe attacking other things that you don't want to do.
Well, they could be, but
they do once you take these drugs.
Yeah.
It could be just something else that's functionally important.
What's that?
It could be just something else that's functionally important.
Yeah, and that everyone believes what it is that you actually still have a problem where the T cells are not able to recognize that cell. Okay, so if you have two problems here, like that this cell is still sort of is stealth for two reasons. One is that it doesn't have markers that the T cell can respond to, and two, it's suppressive. Even if you remove the brakes here, you're not going to get a strong effect. Okay?
So in those 80% of people, the antibody is working, right? Is that the case?
Well, it's hard to tell for sure, but yes, probably in some cases, probably yes. Okay.
It's working, and that wasn't
the problem.
Yeah. So the bottom line here is I want to talk about the two sides of this. So the other type of immunotherapy is completely different and more audacious, but really it addresses the other side. And this is what's called CAR T cells. These are engineered T cells in which we now put in a new sensor that has been designed specifically to recognize a molecule that's on the cancer. It requires that we know something about this cancer, that we know what a unique molecule is, but we put in a sensor that just directly recognizes that. So we're providing, in this case, that T cell function, okay? So in these cases, what we're seeing is closer to 80% response, okay?
And how do you recognize the cancer cells to insert the molecule that will enable the T cells to.
How do you get them to?
You're inserting it in
Oh,
a cancer
yeah.
cell, right?
Yeah, we have different ways of inserting genes into. No, we're inserting genes into the T cells. Yeah, yeah.
You should explain how this happens. You draw blood.
Yeah. You draw blood. This is like a bone marrow transplant. Basically, you draw blood, you can isolate these immune cells, and then you can use various viruses that insert genes to put these new sensors in. It's relatively straightforward. Then you just put, by transfusion, you put these cells back in, and then they are able to survive, repopulate, and then can exert their effects.
For how many cancers do we currently have the set of genes that?
Yeah. I'll get to that in a second.
So my guess about how this might work would be you try to find some molecule that's on that cancer cell and not on most other cells, and find some gene that turns on it, sees that, yes or no.
That's right.
And then if so, it
Kills
triggers
it.
immune response.
Yeah.
Is that basically the only flavor of like you design a specific receptor, or is there some way of altering a more meta logic around like how it combines different kinds of.
Yeah, so that's what we're going to get to. And so let me go through kind of the state of the art. So you take these cells that you modify and put that red molecule on, and you can put them back into the patient. The idea is that these cells will move around and they will identify the cancer cells or normal cells that have this red recognized molecule and start killing them. Okay. So the question that what is amazing is that there are certain cancers, these blood cancers, called B-cell cancers, that for which this therapy was first developed, and just literally like 3 years ago, have led to. I'm sorry, I didn't need to do that. Have led to. Oh, it doesn't matter. Have led to this 80% cure. But let me try to explain why that cancer has worked. So you have a type of white blood cell called B cells. They make antibodies. What's going on here? Okay, you have a type of blood cell called a B cell, and they make antibodies normally, but you can get cancers of those, and your B cells will start expanding, okay? Now, and patients, treated with chemotherapy, but the ones that are refractory have turned to this, and this is the one that's now worked for 80% of the patients, and is going to be the first T-cell therapy, engineered T-cell therapy on the market probably in a year or so. But why does this work? There's a unique molecule that's on all B cells that identifies them, and this will kill those B cells. But it also wipes out all of your normal B cells. And it just turns out that we can live without our B cells. So these patients don't have any B cells for the rest of their life. What we do is we give them injections of antibodies that help them survive normal infections.
So it's important to point out,
yeah.
this is the dumb, simplest thing you could possibly do, which is all that they're putting into these T cells is one sensor, and that sensor just goes after every cell in the body you can find that has the target, and it kills them. There's no discrimination, there's no nothing, there's no computation really. It is just go after.
But the miraculous thing is that as dumb as we're saying molecules are dumb and cells are smart. This is clearly smarter than a molecule, but it's a relatively dumb cell therapy. But the miraculous thing is that it's working in 80% of the people. So finding this particular disease where you can get rid of the other cross-reacting normal cells has shown that this can work.
It's working. Not only is it working, but it's working. There are people who were treated 10 years ago and are still alive. And no cancer treatment has that profile. They typically, except in rare cancers, if you get treated for cancer, you're getting a few months more of life or a few years more of life. 10 years is totally unheard of, and these engineered cells are still circulating in the bloodstream of those patients. So it's a complete change from the way that chemicals and proteins work.
Will those T cells ever decay or go away or something?
So it really depends, but in some of these cases, you can actually get them to stay there and live for decades, just like with having a vaccine in which you're immune to a viral.
Why does it not work for 20% of people?
That is a great question. I mean, it could be that the other side of the coin, that there's immune suppression that's combating against those cells.
So between these two therapies, like if you just give both of them to people with B-cell cancers?
Yeah, that's a great. Yeah. Well, those are the types of things that people are trying to do. But what I want to get at is we really tell you what we're thinking is the vision of what the therapy should look like eventually, and what they need, and it gets at a lot of the concepts that have been discussed here. So let me first, I don't know why, this is on some automatic thing, sorry. So the point that we made is that this is still very primitive, that you have one sensor and it recognizes the cancer cell. But for example, in most solid cancers, lung, liver, pancreatic, etc., that it's really hard to find a marker on the cancer that is absolutely unique. They're often on at least some subset of bystander tissues. And so when people have tried trials like this, we've seen in a few cases lethal cross-reaction, something where one of these T cells reacts with the heart and the patient dies immediately because their heart is basically killed by these T cells. Okay, so these are really powerful, and that's why that control is so important. So what I want to focus on this issue of recognition and how to make it more sophisticated, and it goes forward when I don't want it to, but I think it was part of a sort of a self-running thing. Okay. Anyway, the
there you go.
yeah. Okay. The point is that we want to be able to hack the T cells so that it executes these autonomous programs that allow it to recognize with higher sophistication the cancer. And so I want to first talk about envisioning what would be an ideal kind of cellular algorithm that would be used. And so one of the challenges, I said that when you try to treat, say, like a lung cancer, one of the challenges is that there might be off-target reaction with bystander tissues that share the same molecular signatures. The other is that the tumor itself might be immunosuppressive. So you can imagine, you might want to have a cell that actually is in an unarmed state initially, but when it knows that it's in the lung, for example, it activates, gets primed, and can attack the tumor cell based on that antigen. But then you'd also like them to recognize that they're in this environment and start producing things that both stimulate the immune system, but also start blocking those suppressive things, just like what you were saying. So you can imagine that would be the kind of thing you want to do. And if you had this toolkit, which we were trying to develop, of different sensors, different kind of logic decisions, and then different sorts of responses or payloads, like killing, can we have sensors that have use and logic to decide when to turn on, have two signals to decide when to turn on, as well as reading signals from the tumor to decide to produce things that would lead to blocking the immunosuppression. Okay, so let me tell you a little bit about what we're doing now. Just the first problem of recognition. So we have this classic problem of difficult discrimination between these, where one sensor would lead to killing of both of these things and potentially lethal side effects. One of the things that we've been trying to do is to create cells that have a sort of a gated function where they could only have one receptor initially that recognizes what's called a priming signal. This is linked to the turning on of a gene for a second sensor that's actually the killing one, okay? And then so only when it was first primed by the first signal would it now turn on the second one and kill that. But if this encountered a bystander cell that has the molecule that's normally recognized by the killing, it actually would be off. And so this is actually a really local kind of effect. It's only being primed where it sees the first signal, and then for a certain period of time is susceptible to killing, is able to kill that target.
How reliable are these receptors and gates in actual cells?
Yeah, so that's what we've been testing and trying to develop ways to do that. So we've developed some things where we've taken kind of bystander and tumor tissues that have this kind of distribution of antigens and then injected cells into them. And what we found is that they actually do ignore the bystander tissue, but will kill the thing that has the tumor that has the dual antigens, where the priming one activates the cell to then kill based on the killing receptor. So at least in some of the models that we've done, we've been able to make some simple kind of, and this is again relatively primitive, but simple gates that involve this sequential logic of turning on different systems. This just shows the same results, but what I want to show you here, this is looking at 5 different mice that have these two tumors, the bystander one and the real tumor that has the two signals.
And then this is after 35 days. So normally we inject them, and then we inject the cells. And what you see is that in the same mouse, the bystander tissue grows and grows and grows and doesn't stop. But what we see is that all of the tumors in the same animals are cleared. So there's really discrimination within that animal. So at least, what we're afraid of is we wouldn't want a situation where a T cell gets activated over here and then moves around and starts killing over here, kind of a systemic effect. So it really does show that we can use exactly what you talked about, this local activation. If we can keep the signal local, we can actually get very precise control. So, yeah.
Let's say that we for a second, and this is really wonderful. I just want to, for a second, ignore the biological solutions and say, let's say we made a little tiny camera that went into the body and went around and then had some like sucker on it that could suck up whatever it saw that it consumed as tumorous.
Right.
Ignoring the fact that we can't do that yet, what would happen if we put that in the body? Would the body just say, no, I'm going to get rid of that immediately?
Oh, well, no. I mean, there are issues with that. There are issues about immune rejection and things like that. I'm not going to focus on that. I think essentially this is what we're doing. Okay. I think the thing is that
where are you going with? Well, what I was going with that is that it seems like you're thinking, and what you're presenting is all biological solutions to it.
Yeah.
Whereas it's possible that you just take a different route and non-biological solutions.
Yeah, like a nanomachine that goes. Yeah. Okay. So my point is that how is it that you're going to distinguish the cancer from the normal tissues and really kill everyone? You have to have something that doesn't have like a normal lens. It has to have a molecular lens that discriminates molecules. And essentially, these receptors that we're engineering provide that function. Okay. So I would say that what we're doing is exactly what you were saying.
How easily confused is that, though? It sounds like that's the problem you're trying to solve, is that it gets confused.
Well, we can get pretty good. The heads of these sensors are antibodies, so we can get pretty good recognition. It's the more severe problem is the fact that those molecules that you're recognizing individually can be in many different cells.
Yeah, so tissue's messy is the real problem.
Yeah.
A lot of false positives.
Yeah, but I mean, think of it like you're this is like a Mars rover, and you need to have the right wavelengths and whatnot that you're recognizing. That this isn't a visual sort of thing where someone can look at that and say this is cancer, this is not. You need to know what something that really sees in molecules.
I have a question about the discriminatory characteristics, which is that are they naturally occurring on that particular tumor antigen, or do you have to modify that and then reintroduce it back to the body?
You mean the tumor?
Yeah.
Oh,
the tumor. Yeah.
Well, I mean, in the case of the disease, the idea is to, and I'll get to this. Use the analytics to understand who are the markers that are most discriminatory.
Meaning, I'm wondering, could you introduce such a unique, discriminatory profile to the tumor that then you introduce it to the body?
Yeah. Well, I mean, in the case of disease,
engineer the tumor.
Engineer the tumor instead of the T cell, right?
Well, that's very hard. I mean, there are people, there are things that strategies that try to do that. But again, at some level, you have to pay the price of the recognition to modify only the tumor as opposed to the normal.
That's difficult?
Yeah, that is difficult.
Okay.
Yeah, it turns out that's difficult.
First place, you can't
That's right. Yeah, it's chicken and egg. And what we really think is that these T cells are the best interface for kind of recognizing cells and tissues that have sort of composite, cellular combinatorial differences. Yeah.
Kind of a naive question. Is the way that which T cells kill off cancer cells and other cells in the body, is it just like it induces the apoptosis in the cell?
Mm-hmm.
And if so, do you worry that cancers may have disabled that apoptosis mechanism?
Yeah. So the way it does involve apoptosis, but the way it does is it secretes in a very local way. It binds it and then secretes things that perforate the membrane, and then enzymes that lead to degradation. So it's pretty robust once you get the response.
You can't resist this.
Yeah.
It's pretty severe.
What
if the bystander tissue and the tumor are very?
That's a great question, and we're really looking at that. I think that this is the thing where I'm thinking about.
It's not if they are; they always are.
Well,
sorry, what was the question?
No, not necessarily. I think it gets to this issue of how much more sophisticated can we make these gates in terms of both how many inputs as well as what the length scale of. They're actually kind of this is probably too much detail, but it's kind of a time-based and distance migrated based gating function. So if we can control that timer of how long it's in the prime state, we can shape what the radius of killing will
But
be.
if you've got like a lung cell and then a cancerous lung cell next to each other, like
Yeah, well that could be fun.
is there any way to distinguish between them?
If there were distinct markers, you could. Now, I'm going to turn that around here, which is actually a lot of times tumors are these tissues, and they're very heterogeneous. They're not, we look at like sequence from a tumor, and there might be really unique signatures, but they're only in a few cells, right? And then so you can't kill all. So I think there's some opportunity here too that we're working on, which is that we could, if you pick the right priming antigens versus killing antigens, you can you could probably attack those heterogeneous things. So I would say that the questions you're asking are great, and they are things where there are a lot of problems that you can imagine, but we feel like our basic understanding of cell biology and cellular regulation does give us dials that we think we can tune to adjust those to fit the disease.
I was wondering if there might be a slightly new idea there in the priming of, you mentioned there could be unique signatures in very small percentage of the cells. Yeah. Could you do sort of that cross-cell learning where you prime for an area and you recognize that unique feature, and now you get indiscriminate for some period of time?
So there's ways you could. I mean, that's something that's very interesting, if one of the things that happens a lot in cancer, which is poorly understood, but is a really intriguing idea, is that cancers not only suppress the immune system, but part of what there are certain cells that are involved in presenting and being the initiators of immune response. And if you suppress that, then the immune system can't really respond. But if you can get in and do some killing and then open up that priming of the initiating of the immune response, you might be able to have the body launch its own natural immune response, not these engineered ones, but this is just the Kickstarter. And then you would say, well, if I have a metastasis for this cancer somewhere else, now that would be cleared. And there are definitely effects like that. There's a lot of evidence that even chemotherapy and other things play a role like that, where they just help to kickstart that. So we have ways that we're thinking of trying to promote that happening. But that's fraught with dangers too, because if that happens with in a bystander tissue, you're going to maybe prime autoimmunity. Okay, so it's pretty fascinating stuff.
How good is our current ability to, I guess, our imaging technology, current ability to look at cells and see what is on them and detect the differences between?
Yeah. So I mean, a lot of that comes not necessarily from looking at them, but doing the sequence analysis and proteomics, where we look at what genes are expressed, which ones are expressed on the surface, and now we're moving more towards looking at single cells, so we can kind of get the information about the heterogeneity. And that's one of the things I want to try to get to. Just in the next slide, what we are looking at is really trying to think of these cells. We think of it as being little nano robots that are built to recognize a whole set of different signals and to integrate them and make decisions and to carry out these particular functions. And as we develop new ways of modifying these sensors, linking them to particular decision-making circuits, and then whatever the payloads are advantageous, we think we can make a lot of different types of programs and try to fit them to the needs of particular diseases. So a lot of what we're trying to do is to cure certain specific diseases, but also at a much more foundational level, ask whether we can develop the toolbox or the language of programming both cellular recognition and response algorithms. I think an important issue is that there's a lot, and maybe some of you have thought about kind of bioinformatics and analytics of health records and information about different diseases, and there's that's very interesting in terms of collecting data about different tumors. Right now, of course, that information really is useful as a way of stratifying patients and classifying them and saying who might correlate with a response to a certain drug or other things like that. But what we think is really exciting is that when you combine the ability to have precision informatics and analytics with precision cell therapies, that's the perfect match. You can turn that disease analytics into actionable therapeutics and really have the promise of true precision medicine, okay? Because right now it's more old style medicine treating the information that we have. So if we have this toolbox that really allows us to customize the behaviors of the cell, we can go in, take the information we get about tumors, understand what are signals, inputs that we should try to recognize that have the most discriminatory power, and then what are the things that we need, what are the countermeasures for this particular cancer in terms of what it's producing and other things in terms of its immunosuppressive environment that would help to really overcome those issues. And then if we can build those, then we can really address the disease in a precise way. So I think that's it for me. Happy to answer more questions. Yes?
How hard is it to design a cell right now, or to engineer a cell, and what's the process that you use?
Yeah, what's the process? So, I mean, that depends on the scale that you're talking about, but I'll tell you, when we're, let's say, just putting in, like the current generation therapies where you put in this new sensor molecule that recognizes the thing on cancerous B cells, what you do is you harvest the patient's immune cells, and then what you create a gene, you synthesize a gene that makes this sensor molecule, and then you actually put it into a viral vector. It's actually based on HIV, but it's non-pathogenic, and that's because HIV will infect T cells. That's why there is the disease. But they will infect T cells and integrate, but that it doesn't replicate, so it's not toxic. Although it gets people nervous when you talk about it. But in fact, one of the first New York Times things was not focused on the actual meat of it. It just said, doctors use HIV to help, program the immune system. But that's really kind of a side issue. And then it takes a matter of weeks to grow those out, and then you can put them back in.
And then you inject them into the person.
Yeah.
But how do you how do you program the logic?
Well, right now it's very Stone Age that we have these sensors, and we know kind of how they can turn on different genes, and we can start building circuits. But it's what we really would like to have is a much more modular and extensible system that we're really just beginning this kind of work. But if that's the case, if we have that language, then it opens up to people, who you guys can sit here and you can program things because of the languages that you use, right? And then you can really look at all these different, the information and use it in some way. So, we're building out very slowly.
I think it's telling that the cell therapies that are curing people's blood cancers right now consist of one gene being put into their T cells, and that gene has no control. It's on all the time. That's it. It's that simple. There's no logic at all in the way that we've been describing it.
Right.
No even simple circuit like two genes, and when the first one gets activated, then it turns the second one on. That's so dead simple, but even that hasn't been implemented yet. So we're at the very beginning.
Going back to your earlier slide, why do you think this is similar to face recognition or any of the other algorithms we use?
Well, I think it's more aspirational. That I think that if we could, it's in terms of things like self-driving cars. You need to provide this. You have to provide the cell with the information it needs to be able to navigate this very complex environment. Okay, I think we've done some things like that at a small scale, but it isn't at the sophistication that we're talking about in those other cases. But I think we can get there. You guys first.
In terms of the actual mechanics involved in building a cell design toolkit,
Yeah.
what do you need right now? Do you have all the information you need, or what's currently missing?
What we tend to do is we sort of borrow components that exist in natural things and modify them and kind of cobble them together and ask, when they allow us to sort of reconnect components in a network in different ways. That's, and when we kind of find something that works, we sort of repeat that in different ways. But
so is it that you're looking for different gene sequences that do different things, biology background.
I think the thing is that like biology tends to be very descriptive. There are a lot of components out there that do certain things that, let's say that a promoter will turn on a gene in response to hypoxia, like a low oxygen environment, which is often the case of a tumor. How do you actually take that and integrate it in a non-natural way into a circuit that would be useful for this kind of discrimination? Is that we're always constantly looking for those things and realizing that we can put it together in different ways to make new circuits.
Where does sort of machine learning and simulation come into your job and your team? And
so
how do you think about that to determine these? And
sort
of a second question is, why have you not said the word CRISPR?
Okay.
And like, is it a branding thing? Or
No.
Is it a copyright thing, or like, is this what we're talking about?
No, it isn't. So it's not. Okay. So you asked two questions. What was the first one?
Machine learning.
Machine learning and simulation.
Yeah.
We talk about these things on a protocol, being able to communicate. We can simulate those things. We can.
Right. So I think the most obvious place right now is on the side of kind of trying to take data out there and understand what are the actionable sort of algorithms that are both specific but maybe as simple as possible.
What three markers distinguish one tumor from the healthy surrounding tissue?
Not in the what is the T cell gene expression that will bind directly to that marker?
Because if you told me the three things that A plus B not C completely distinguishes or best distinguishes a lung tumor from the then Wendell could build the components into the engineered T cell that will recognize A and B.
But it could be, you could use the T cell information too. That you can take, for example, data from a cancer patient who does respond to a checkpoint inhibitor, and the T cells there might have a certain signature. And you could say, okay, this is, maybe what we need to do is turn on this set of genes to make our T cells look like those.
So if you sort of sort rank the roadblocks towards wider reach of this technology, one of them is recognition of unique markers.
Right.
What's 2 and 3 if you had to?
Well, the state of the T cells is very important. Yeah, so,
what do you mean by state?
So Michael talked a little bit just at a simple level how they can be activated or suppressed.
Sure.
So things like that, and we don't even fully understand what those states are.
That the second question, which is like, what's the T cell design that can most accurately bind to the?
We're still aggregating parts and trying to build programs that link the parts together. So how to do that? There's so many different designs you could come up with a finite set of parts and ways to link them together that knowing how to demo those in silico before we build them, because it's slow once we build them.
But that to me sounds like
that's a huge thing. That's totally machine learning, especially because the parts don't always work predictably. So instead of getting upset about that, trying to figure out how to harness it and to predict when they'll work well.
But whether we would have the capabilities to sort of do something actionable on that information is still, that's what we have to develop.
So what's the third roadblock then? If those are the first two.
I think those are both huge roadblocks, and that's all there is.
Yeah.
Wait, you didn't answer the CRISPR question.
Oh, yeah.
That was a dumb question.
So we use CRISPR, but we don't use it for all of these things, and most of the things I talked about don't involve CRISPR. CRISPR is one way to modify genomes, these HIV-based vectors are another way. They have different strengths and advantages. So CRISPR is really more of a text editor and less of I think it's definitely good for this idea. I mean, it has issues of kind of correcting small changes that are in genes that lead to hereditary defects.
Drop 3 paragraphs in, you use the modified HIV.
Yeah.
It's easier and it's so much more efficient.
Than CRISPR.
Than CRISPR is
so it's a benefit to do hyperlocalization.
Wendell and I tend
to do what?
To like hyperlocalize.
We tend to be agnostic about how to do the genome editing. We're more on what do you want to do with it. So there's been a lot of craze around CRISPR. If you read carefully, the applications people talk are relatively few in number, and they're kind of modest. The only unmodest one I've ever seen is gene drives to kill malaria, to kill the mosquitoes that deliver. That's interesting and a little dangerous. But we think this is going to be one of the major application areas, not just for CRISPR, but for all of the genome editing technologies that people are working on now that haven't hit the press yet.
I guess I still don't understand. You said you use the HIV vectors for paragraphs, which I'm guessing you mean rewriting tons of.
Dropping new code in,
Yeah.
new genes into the genome.
I understood that it's smaller than the CRISPR edits. Larger?
No, CRISPR edits.
it's much
CRISPR is
smaller
much
and
more
smaller.
modest changes than you can. If you want to put 3 new genes into a cell,
No, you still have a problem getting the CRISPR in, so it has different
One
problems
question
in
at
it.
a time. So if you want 3 new genes into a cell,
then HIV is the way to go.
but if you want to modify a gene, then it would be
CRISPR is the way to go.
Yeah. I mean, there's a lot of hype associated with CRISPR, but what?
They're doing pretty good on branding.
Yeah, why is that? Tell me why you think that's.
I don't know, but like here's tell the difference. That's my fault.
Yeah.
They're running it New York Times.
It is interesting.
But you see that the promise of genome editing is, I think it's huge, but I don't think that the applications have been built out in the popular press. It's just basically like, oh, now we can do this, the world's going to change. And if you ask, what do you actually want to change?
Yeah.
Then the answer is...
It's more like, CRISPR allows you to have a text editor that you can debug this line of code, whereas we're talking about trying to just, write new code. And how do you do that? It's not at the letter by letter level.
But the two questions, the two sort of ranked problems you asked were mentioned were related to that, though. It's like understanding detection, which, given the ability to edit code, you could iterate through the possible.
Sure.
That's right. We think that there are plenty of tools, and people are developing new tools all the time for doing genome editing, and that's a fantastically enabling thing. But we're more focused on what are you going to do with that? So now I've got a great way to do whatever I want to edit the genome.
But we would love to be able to introduce a new chromosome into the T cells that gives us a big, sort of payload to put in information. Things like that. Okay. And if you could do that with CRISPR, that's great. But that's the bigger payoff in our mind for the things we want to do. Yeah.
So one of my friends that works in a similar field to this is very fond of saying that nature is always fighting back when they try to do directed evolution, or when they try to create new things. So, like, you're taking these T cells, they're not dumb systems that are empty shells, right? They're, let's put it for better, I have no better terms, but there's millions of lines of code already inside them.
Yeah.
So you're trying to add in new things. That seems like an extremely complex problem.
Yeah, I think so. So that's a great point. I think it's a question of kind of one thing you have to understand is that immune system. I mean, CRISPR is an immune system, and it has a certain modularity to it because it's meant to be able to be retargeted. Okay, and I think that's a property of a lot of immune systems. These cells are they have a core sort of response, and then they also have some modular components that change, and that variability is part of hardwired into them. So I think that this is a platform that if you find the right connection points, you actually can work with nature, okay, more than most other cases. And some of these things with respect to those initial sensors and plugging them into the right way, this works best when you kind of ride as much of the natural response as possible.
The directed evolution connection is that the cancer will fight back. So if you
that's true.
node in the cancer, it'll definitely find another way to do what it was trying to do anyway.
But that's where we actually think a cell can actually respond
If I recognize 3 things about a cancer, and then by the numbers, it can't change all 3 at the same time. Because the statistics of that are that it's such a rare event that it won't happen.
Yeah.
You'll be tickled to know, given the code thing, that sometimes when we put code into a cell that already has millions of lines of code, it gets silenced. There's a code silencer in cells that silences if you put it in the wrong place.
Nice. You just regress all the proteins.
It's a complicated.
And it keeps that part of the genome all wrapped up so that the genes can't be read. It's an FU to your new code.
Yeah.
Can I ask about the observability of the T cells? Let's say you engineer them, and you place them in the body. How much can you track them and then get information?
So you can track them pretty.
So you're going to erase the chunk. You have to walk around.
Oh, that comes off. Wow.
Yeah.
That's a lot of. So you can track them pretty well. You can take blood samples, and now with kind of sophisticated sequencing methods or mass spec methods, you can see if you can identify they have distinct markers, genetic markers, because you modify them, so you can see where they are, what state they're in, and so
So
forth.
you can tag them. How do you gather success and fail of how they're doing?
Yeah, well, that's. I mean, you would be looking for whether they would be expanding and where they are and things of that
So you
sort.
see how many kills they've had?
Their kill number is related to how many when they kill, they divide.
Yeah.
So they.
Oh, okay. So there is.
Oh wow. It breaks down.
But that's.
It's just if they've been successful at killing, that often in a perverted logic we're talking about causes them to expand themselves.
Oh, yeah.
When they.
That's yeah. I mean, I can, since you want to see killing, I can show you some movies. Well, okay, so wait, this is a different cell that we've engineered. This is a T cell recognizing cancer cell. This one actually is controlled by a drug. So without the drug, it just holds on to it, but it doesn't kill it. And then I didn't talk about it, but it's a way to give user control over this. But this is the one on the other side is the cells with the drug added. So the green cells are the T cells, and what you'll see is okay. So this one holds on to here, and then immediately you see this kind of blebbing, and this dye is indicative of cells dying because it goes in, and so it pops that guy. And then once that's well on its way to being dead, you see it reorients those sensor molecules over here, and then starts attacking this guy. And you'll see by the end of the movie, both of these are kind of really just blobs with a lot of blue, indicative of the death.
It just turns to its left.
That's amazing.
It is. It's really amazing, and we can steer these in different ways. But of course, they can kill your normal
A typical
tissues.
cell can get thousands and thousands of kills. No.
This is about 20 minutes. Yeah. This is about 20 minutes. Yeah.
That's 20 minutes, okay.
You had a question?
Oh yeah, I was just curious because a lot of what you guys have been talking about is killing cancer cells.
Yeah.
You were mentioning the other side, which is like autoimmune
Yeah,
disease.
yeah.
Has there been successful work or experimentation on treating that and getting the T cells to back off?
Yeah. So one of the things that we're interested in doing is these are the cells that kill, and then as Michael said, there are these suppressive ones called Tregs. One thing is trying to say, could we engineer the different cells to recognize, say, let's say that you have diabetes or some other, say a kidney inflammation or something. Could you redirect these immunosuppressive T cells to go to the kidney and change that environment? The other thing is we're actually also trying to take sort of more naive T cells, actually engineer them to produce things that are immunosuppressive in response to recognition of a tissue that is associated with autoimmunity. But this, if you can do this, there's a lot of great things that could happen too. You could imagine that when you do an organ transplant, which would normally be rejected and require a life of systemic immunosuppression, which has lots of problems, that you would actually put a companion protecting T cell in it that would buffer the immune response against it.
I think we should eat. What do you
I think there was one
Well,
question right there.
After this way,
Yeah, I had to answer more.
Brian, you had a question right here.
Oh yeah, you said that you engineered the T cell to have a, kind of like if it sees this antigen, attack it, and there's no kind of more complicated logic. Outside of in other types of cellular engineering, are there already examples of kind of more sophisticated logic, or is that kind of state of the art right now?
In terms of sort of our own engineering?
You started as logic gates.
Well,
are there people out in different contexts where they've built these more sophisticated logic gates? Is there prior art on that, or is
mostly in bacteria, I would say. And what I would if you're asking about natural systems, almost all biological systems do have lots of layers of gates, and we're really just trying to kind of harness that capability. I would say, at least in this sort of arena of immune cell engineering, this is one of the first cases. It's mostly let's say, so bacteria, people have done this, but even say like stem cells, where we're trying to differentiate them into heart cells or something like that. There's a lot of effort to try to figure out how to push them there, but not how you decide when and where they're going to turn into those things. So a lot of these things that we're talking about here can be used for that kind of.
The best synthetic circuits I think that have been made to date are in, as you said, bacteria by a guy that Wendell hired, who then hired me and then left and went to MIT. He's built named Chris Boyd, who's built some tremendously complicated circuits in bacteria that aren't very useful right now, but show that you really can construct something complicated with 50 or 75 parts and it'll work predictably.
What's an example of that? 50-part machine?
A 50-part machine can turn bacteria into photographic film, where if they're exposed to light, they turn one color, but if they're not exposed to light, they don't, and the ones at the edge can do something different.
They've done some edge detection and other things. Sort of,
At a cellular level.
Yeah.
Are you guys using I don't know, it's called Cello? You heard of this?
Yeah, that's what he's talking about.
That's the same guy.
Okay, cool.
Yeah.
So that's like Verilog for cells. You guys should take a look at it.
That's very good. It's cool.
Yeah.
So what are the applications of it?
I think they're struggling to find compelling applications.
The problem is that a lot of these things, I mean, we can talk about what, the applications, the kind of framework for modeling these things and designing them. And mostly the problem is that things aren't like aligned properly, right? And so you can make you can design really complicated bacterial circuits, but it's not clear what the applications would be.
He's becoming interested in human cells for that reason, because you're sort of at an application dead end with bacteria. I'm sure cool things will come about, but this is...
How are we at a dead end with applications of bacteria, given that we've just figured out all these benefits associated with the microbiome?
The microbiome, there are going to be a lot of things we can do, but a lot of the work to engineer bacteria that Chris has done and others have been built out in bacteria that you wouldn't want to engineer. So he's beginning to port what he's done into microbiome organisms, where there will be huge opportunities.
Yeah, so you guys kind of answered this, but like, what tools and technologies have enabled these recent breakthroughs in both studying the microbiome and this stuff? And what future things will really bring all this together and hit the tipping point or whatever?
Yeah, I mean, so obviously basic information gathering from sequence and observing cells and studying them have led to us having even this kind of framework to think about them in this way. And I think that we're in this transition stage in biology of moving from just gathering information to asking kind of how the systems work, how do they actually, what's the logic behind them, and how can we manipulate them.
Let me try one.
Yeah.
So this is actually Wendell's argument that I'd restate, which is that engineering thinking once it permeates the field, it opens stuff up. Once engineers permeated chemistry, that's when you've got this big explosion of chemical drugs. Once engineers permeated the study of proteins, that's when you got protein engineering, and Genentech and Amgen were founded around 1980 on that technology. And now that engineers are beginning to permeate the study of cell biology, people like Wendell are just beginning to build out a basic science of how to do engineering on cells rather than trying to pick them apart one piece at a time, as people have been doing laboriously for ages. So it's a different mindset. Like, you guys get the engineer mindset because that's how you were trained or wherever you came from. But people who study these systems, it clashes with their logic of let's just take one part out at a time and see what happens.
You mentioned earlier that one of the roadblocks was detecting the difference between tumor cells and bystander cells.
Right.
What is preventing really what I'm asking is, what would really accelerate your ability to kind of be able to do that differentiation? Do we already have all the genomes of all of these cells and now we just need to analyze it, or is it that we have to go gather a lot of information about all these cells?
That is a great question. I mean, we do have a lot of information, a lot of sequence information from different tumors. And I have a collaboration where we're going through all this data with these kinds of questions in mind. I think, what a lot of, and there are signals that are coming out, first of all, but you have to think about which are the ones that you really could recognize with sensors that you engineer. But in addition, there's a lot of noise, as you might imagine, where, and part of that is, the heterogeneity of cancers. You don't know, you have to parse different, that something might be classified the same way, but there are different subclasses. And then we also really don't know that much about, at the single cell level, what the sort of geography of a tumor is like and where the signals are and so forth. But that's starting to come. But how I think, again, most things have been focused on correlation with outcomes with treatment, and I think just when you start doing the machine learning, that's not the metric we want to use. We want to look at the discrimination, and then also it is a challenge to think, how do you look at the normals? Actually, there's a lot less data on the normals, and also, do you want to compare against normal tissue cells everywhere, or if you're going after a blood cancer, do you want to also just focus on comparison between the cells that are closest in sort of their lineages? So there's some interesting real world problems, but I'm pretty optimistic.
Last question, and then we're
Okay.
wrapping up.
Sure. Is as far as like figuring out what these markers are, do you like look at it structurally? Do you look at like genomics? Like is UCSF do like IBM or?
Yeah. So I mean, it's less. I mean, all of these things play some role in that, like a marker that's at the membrane that has enough sort of bulk to recognize is important. But for the most part, it probably can be done at the sort of genomic or sequence level. And there's, as I said, there's a lot of data, but new techniques are coming into play in terms of how to recognize them. Okay. Well, thank you guys very much. This is really, really great.